Welcome to today's webinar, Real-World Considerations in Pharma for QSP Modeling, presented by Kyunghee Yang, Principal Scientist, Viera Lukacova, Chief Scientist, Scott Siler, Chief Scientific Officer, and today's Q&A moderator, Brett Howell, Divisional President. Our goal for today's webinar is to educate and facilitate a conversation on how our scientific solutions can help you and your organization. A few housekeeping notes before we get started. We take your privacy rights seriously, and by attending this event or participating in the Q&A, you allow us to catch up and follow up with you for additional information. Our goal again is to inspire conversation with scientists at all levels of pharma R&D. So if you have a question at any time, please pop it into the chat panel or the questions dashboard, and at the end of the presentation, we'll be sure to get your question answered. And if you need assistance at any time during the webinar, use the hand raise icon and our staff will assist you. Without further ado, please welcome Kyunghee to the virtual table. Take it away.
Thanks, Arlene. So welcome everyone to today's webinar. So, and here's today's agenda. I will start off with a quick introduction to our group and QSP modeling, followed by a presentation on PBPK modeling to predict drug exposure by Viera Lukacova, and development and use of SimPops, simulated populations by Scott Siler. And then I will present case examples where QSP modeling was used to address real-world questions in the pharma drug development. And at the end, we'll have time for panel discussion. So with that, let me start with introduction. So here at Simulations Plus, we have expertise in cheminformatics, physiologically based pharmacokinetic modeling, quantitative systems, pharmacology, and toxicology modeling, and pharmacometrics. So we have multiple software platforms that cover these different areas, and you can license our software to get access and use it on your own, or we can provide consulting services leveraging our software platforms and expertise in modeling and simulations. And we can provide support at any stages during entire drug development processes. And today, our webinar is focused on PBPK modeling and QSP modeling. So a little bit more on QST modeling. So we rely on established QSP modeling methodologies to develop the specific QSP or QST models for a certain disease area, leveraging existing preclinical clinical data to develop and construct the model and validate the model. And those models then can be used to support development of a compound for an indication. So we have two QST, like toxicology platforms, to predict drug-induced liver injury and kidney injury, and two release QSP platforms to predict drug efficacy in non-alcoholic fatty liver disease and idiopathic pulmonary fibrosis, and QSP modeling of radiation-mediated injuries in development. In addition to that, we also provide customized services where we can develop from the scratch the custom QSP or QST models to meet your needs in a specific disease area. And when it comes to QSP, it integrates the known pathophysiology and drug-specific exposure and pharmacodynamics of that drug to predict efficacy. And mechanistic mathematical representation of underlying biochemistry describing pathophysiology is the key foundation of QSP models. And within this QSP model platform, we also have simulated population or simulated patients with variability in these pathophysiological processes, as well as disease state and severity or progression. So this platform, QSP platform, allow users to really predict the efficacy at the population level with the population variability in responses. And you will hear from Scott Siler about our SimPops development and use in the later section today. To predict drug efficacy, we start with drug exposure, and the drug exposure at the site of action is predicted by PBPK modeling. And you will hear from Viera about the PBPK modeling approaches soon. And we combine drug exposure with its PD effects or mechanism of action, which really dictates how it interacts with underlying biochemistry and pathophysiology of a certain disease to provide treatment benefits. And since there are multiple targets it exists for a certain disease, QSP model really needs to be flexible to provide the ability to represent different therapeutic targets and their subsequent treatment efficacy. So with that, at this point, I will turn it over to Viera to go over the PBPK modeling section.
Thank you, Kyunghee. We'll start at the beginning, right? Start talking a little bit about the PBPK modeling and why it is such a great platform for predicting or characterizing the concentrations at the target site. The picture on the left is a general schematics of a PBPK model. And many of you may be familiar with this schematics since you might have seen it in publications, in different papers and so on, and it outlines the layout of the different tissues in the body. And so the PBPK model is basically a compartmental PK model where the compartments represent specific tissues in the body. And with that, they are described by a physiologically relevant sizes, like volumes of the tissues, blood flows, highlighted by those blue arrows in the picture, lymph flow, which may be significant for certain types of molecules or maybe significant mechanism for certain types of molecules, enzyme transporter expression levels, general composition of that tissue, which affects how compounds of different properties would be distributing to that specific tissue and so on. And this type of model, and we'll look a little bit more into details of what is happening within those tissue compartments, then allows to better characterize distribution of the compound to the tissue when depending on the mechanisms and the type of the compound that you are trying to model. Next slide, please. So when we talk about small molecules, right, this is where the distribution between the blood and the extracellular tissue space usually is not a major concern. The pore sizes within the between the cells in the endothelial wall are big enough for small molecules to freely diffuse, right? So we usually have a very quick partitioning between the blood and the extracellular tissue space. But then we might need to account for additional mechanisms to account for distribution of the compound between the extracellular space and the cells. So either transporter-mediated uptake and efflux, or potentially slow passive diffusion through the cell membranes to get into the cells if the target site or target protein is within the tissue itself. When it comes to biologics or larger molecules, this is where the rate limiting step or the barrier moves more towards that endothelial wall, right? And the bigger concern is for the compounds or for these large molecules to actually permeate into the extracellular space in the first place. The molecules, especially we are looking at the monoclonal antibodies, the pore sizes are not big enough to allow for any significant diffusion directly. So other mechanisms for the distribution may need to start playing a role. Things like endocytosis process, which brings the compound into the endothelial cells, forms the endosomes and lysosomes. Of course, within these, the compound could start degraded. And again, there is a protective mechanism for monoclonal antibodies binding to the FcRn receptor. The compound that is bound will survive. And when the vesicle or the compartment opens up on the surface of the endothelial cell, the surviving molecules will get released back either into the plasma or into the extracellular tissue space. And the additional important mechanism that we may need to consider for these large molecules is target-mediated disposition, or how binding to the target actually contributes to degradation and elimination of not only the target protein, but also the antibody itself. And this type of molecules are also the ones where the lymphatic flow becomes the more significant aspect, or the drainage through the lymph, more significant aspect for the distribution of the compounds. Next slide. So looking at these different mechanisms and really breaking down the tissue space in the individual subcompartments really does provide a good platform, good vehicle, or good model for simulating not only plasma or total tissue concentrations, but looking also at the concentrations in those individual subcompartments, whether it is extracellular space or intracellular space, looking at total or unbound concentrations, whichever is the most relevant concentration for the specific either a pharmacological or toxic effect of the molecule that you are looking at. So now you are probably asking yourself, well, how well can we trust those predicted tissue concentrations? And of course, the validation, the most robust validation would be by comparing these predicted concentrations with direct measurements of concentrations in the tissues. This is typically not feasible, especially in the clinical setting. Right? So some other approximations or other strategies for validating or gaining more confidence in these predicted concentrations needs to be employed. And one of those might be using preclinical studies. It is possible to measure some of these concentrations in the animals, use those to evaluate or making sure that we are accounting for correct mechanisms that are affecting the specific compound of interest. Right? And once we understand, once we have confidence that we are correctly capturing or correctly describing all the mechanisms responsible for the compound's distribution, that gives us more confidence in translating it into the human and in those predicted concentrations in human. In addition to that, there are possible indirect verifications with in vitro data. Right? For example, if the compound is, if you are interested in liver concentration, right, and the compound is metabolized in the liver, and you measure the in vitro, unbound in vitro Km value, and this unbound in vitro Km value is able to correctly predict any nonlinearity in the in vivo clearance, right, that again gives you more confidence that those concentrations are correctly predicted because they are relating to that Km value as you would expect. So the model applications, of course, one of the big applications would be starting with the first in human predictions. And a lot of these discussions were in doing the predictions either from in silico data or from preclinical studies, doing those initial predictions of the concentrations for very early evaluations of the potential pharmacodynamic or pharmacological responses. But that's not where the utility of these models ends. Another big area would be exploring different dosing scenarios. Even if you are dealing with a case where maybe the plasma concentration is the most relevant concentration for the pharmacological effect, and you have already clinical data for the plasma concentrations, the models would still be useful, allowing you to start exploring different scenarios. So you don't have to do clinical studies for every single scenario, every single administration type, but you can start predicting what would happen with the different doses, different dosing regimens, and so on. And another big area is extrapolating the effect or extrapolating the PK to different populations. There are populations in which, for ethical or other reasons, you cannot do the clinical studies just to explore what is happening with the compound, right? Just to explore some of these effects. Let's say evaluating what might be happening in children, right? Starting predicting the PK in children or in elderly subjects or in patients with various conditions. And the physiological model is really a great aspect or great methodology, again, to start predicting or start translating these concentrations and the PK from healthy adults into different populations of interest, because once we understand or when we know how the physiology or the specific critical part of the physiology differs between the healthy adults and the population that we need to simulate, that we need to predict, we can incorporate those physiological changes in the PBPK model and start predicting how those changes in physiology would be affecting not only the PK, but with the link to QSP, QST models, also how it would be affecting the pharmacological response. And here I will start with this very brief overview of PBPK models and why to use PBPK models for these types of applications of predicting those exposures. I believe Arlene has a quick poll questions for all of you before we proceed with the presentation.
Are you currently using a commercial PBPK platform with your QSP modeling? And we'll give you just a few seconds here to cast your vote. All right, we'll go ahead and close the poll and share those details. And now we'll turn it over to Scott Siler.
Thank you, Arlene. So it's my pleasure to share with you all a little bit of information about our simulated populations, or SimPops, and the way that we generate them and the way that we use them, ultimately setting the stage for Kyunghee to show some really nice examples of all of the above. So in the next slide, what we have here are a number of different kind of visual aids representing kind of some of the various applications, utilities of these simulated populations. I think conceptually it makes a bunch of sense in that, our populations are diverse, and rarely does one population respond the exact same way to a single treatment. And hence, as we're simulating the response to a treatment, as we would with QSP modeling, we need to as well incorporate that diversity of responses. Same on the quantitative systems toxicology side. We need to be able to predict in particular on that side, kind of the rare instances of toxicology that are very important considerations in drug development. So we've been practicing kind of long established methodologies in order to do this. And a couple of particularly useful aspects here, I think, are the ability to discern, for instance, responders from non-responders for a particular treatment, as well as, depending on where you are in a clinical development pipeline, first in human versus phase 1B, phase 3, perhaps you're targeting different subgroups of patients. And so by providing a large SimPops that really includes a diversity of patients across the pathophysiologic and clinical spectrum, it enables users to select specific subsets or sim cohorts that really allow you to simulate a specific clinical trial throughout the pipeline. Again, we develop our SimPops with all of this in mind, that versatility, that utility. And we've gotten some fantastic feedback along the way. If you advance one more, please. You'll see this quote from one of our users. I believe this was specifically referring to our NAFLDsym platform, where the SimPops are the crown jewel. In that particular platform, we have, I think, over 1,700 varied simulated patients, i.e. a very large SimPops, with, again, a large diversity amongst the pathophysiology and the clinical distribution. So if we go to the next slide, I'll kind of introduce you to, again, these well-established practices to which Kyunghee referred, the methodologies for generating a SimPops that we're applying for yet another platform, and this is IPFSim. And so IPFSim version 1A, you may have heard, you may have seen our webinar, seen our announcements, just released. And so as we made the SimPops for this particular platform, the formula we followed is, if you'll advance, you can advance two more, how about? So what we did was we went into kind of a triangulation, if you will. A, within the model, what are the key processes representing the pathophysiology? B, within the literature, what is the range of responses or range of characteristics of the various aspects of the pathophysiology? That last part, the range, is critical. And then what are the parameters that kind of correspond to those specific ranges that you would see in those in the pathophysiology? And so you can see in this table that, this with idiopathic pulmonary fibrosis, some of those areas included immune cell recruitment, alveolar epithelial cell, apoptosis rates, the extracellular matrix, or kind of the critical component of fibrosis, the turnover rate, the synthesis degradation, and progression rates. And so we went through and did all the combinatorics, generated a wide array of simulated patients. And if you'll click two more times, there we go. You can see what we were able to generate, again, a wide array of patients across the pathophysiologic and clinical spectrum. And then we set about validating these patients by additional comparisons to clinical data to make sure that across all of the pathophysiology, everything is indeed within the range of what has been reported, what has been observed in the actual, in this case, idiopathic pulmonary fibrosis patients. The final step is really crucial in as much as it allows some confidence to be established that not only can you generate kind of the progression aspects of the disease, but also the response to treatment. And that is to subject all of these same new simulated patients within a SimPops to the standards of care and to ensure that, in fact, they're responding to treatment appropriately. In this case, with idiopathic pulmonary fibrosis, the two treatments that are most widely available to patients are nintedanib and pirfenidone. And certainly we have gone through all the validation protocols and to ensure that, again, this large group of simulated patients, our SimPops, are in fact in alignment with both the progression and the response to treatment. So to summarize, walking through our understanding of the disease, the range of the pathophysiology, how the combinatorics of each of those ranges and each of those pathophysiologic elements, generating a wide array, simulated population of simulated patients that allows us to predict a wide response. The next two slides, I'll show you a little bit of the validation because I think this is really critical. In idiopathic pulmonary fibrosis is a really nice imaging methodology, this HRCT, that allows us to quantify how much of the lung is in kind of specific disease states, if you will. In this case, there are kind of two areas that are really, three areas that are really crucial. One is the normal parenchyma, i.e. unaffected tissue. Another is honeycombed. So this is the tissue that has so much fibrosis that it is completely no longer functional. And the third is kind of the in-between, what's known as the fibroblastic foci. This is also imaged as the reticular opacity. And so we're able to utilize the clinical data to understand the range of patients with allocations, volumes, percent of their lungs in each of these categories. And so if you'll build out the rest of the slide, what you'll see is really nice alignment with our simulated population of IPF patients. Those would be the individual red dots, each dot representing a single patient. And then compared to the clinical cohorts, where there's a mean and their standard deviation, and you can see again that they're nicely aligned. You can also see, as you compare the two clusters of dots, that as we consider patients that are early in the disease spectrum versus those that have progressed and are further along, that you can see that the volume of the disease state, be it the fibroblastic foci or the honeycombing, is increasing over time, and you can see that distribution is kind of shifting up, reflecting that progression. Again, nice validation that we've have an appropriate range of both the honeycombing, the fibroblastic foci, two critical both pathophysiologic and clinical components. And then if we take that to the next slide, perhaps the most important aspect in terms of evaluating efficacy and understanding the IPF patients are the actual respiratory, the measurements or the outputs of respiratory function. In this case, the forced vital capacity, or the FVC, or the DILCO, which is a measure of the diffusive capacity of gases, in this case, carbon monoxide, across the alveoli. And so again, you can see then that our SimPops, simulated patients within the SimPops, line up very nicely with the range of FVC reported in the literature. Again, you can also see the shift as you move from the early progression patients versus the later progression patients with a declining respiratory function indicative of the disease progression. And then same thing with the diffusing capacity, the DILCO, a worsening over time with all patients really nicely lining up with the data. It should also be noted that there are some patients obviously falling just outside of that range of data, and that is very much intentional. It allows us to explore certain hypotheses of patients, whether they're relatively early in the spectrum, relatively late in the disease spectrum. Again, enables selections of sim cohorts to determine if maybe an early patient would be more responsive than a late patient, et cetera, et cetera. So that more or less summarizes our approach, the utility of our SimPops, sim cohorts, and with the example coming from the IPF Sim version 1A. Now we'll have a poll question, then we'll turn it over to Kyunghee to show how we've used some of these SimPops to make some real world predictions within the context of pharma. Go ahead, Arlene.
Thanks, Scott. Do you, in our audience, heavily utilize virtual populations within your QSP modeling application? And again, we'll give you a few seconds to go ahead and make your submission. Then we'll bring Kyunghee back into the mix. And don't forget, if you have any questions, go ahead and pop them into the questions or chat panel on your dashboard. We'll go ahead and close the poll now. And here are the results. And we'll turn it right back over to Kyunghee. Take it away.
Okay, thanks, Arlene. So for this section, now I will present specific case examples where QSP modeling has been applied to address real-world questions in drug development. And from our experiences, QSP modeling has been applied at different stages of the drug development pipeline. In preclinical stage, it has been used to evaluate a specific target or to translate preclinical findings to humans. In early clinical development, QSP modeling has been used to inform the dosing protocol design, and also to provide some mechanistic insights for some observed drug effects from early clinical trials, and also to predict some long-term histological outcomes based on early serum marker responses. And it has been utilized in late clinical trials or development stage as well to identify some sub-target populations that would benefit most from the treatment. and also to predict responses to combination therapies with different targets. So I will share 6 specific examples in next few slides. And today I will focus on application of the QSP modeling rather than methodology, since methodology has been presented multiple times previously. So in our first example, so this was the collaboration with Pfizer, which used DILIsym, a QSP modeling platform that representing NAFLD-NASH pathophysiology and like therapeutic target and efficacy. In this project, the QSP modeling was applied to evaluate AMPK activation pathway as a potential target for NASH treatment. So we have represented AMPK pathway, like activation pathway and its effects within DILIsym. And we ran simulations using SimPops with varying levels of AMPK activation. So in this case, this is more of a sensitivity analysis type of approach for target evaluation without any specific data, drug-related data. And simulations showed that activation of AMPK pathway could lead to reduction of steatosis and also lipotoxicity, suggesting that it could be a potential target for NASH treatment. And also simulations showed that in a subset of patients, the maximal liver fat reduction was not achieved during the 12 weeks of treatment. So it provides some information guiding clinical trial design, really suggesting that the clinical trial has to extend over beyond 12 weeks to achieve maximum steatosis reduction effects in NAFLD and NASH patients. In the next case, this was actually a collaboration with Genentech using NAFLD-Sim. And this modeling was used to really evaluate the mechanisms responsible for drug effects observed in phase 1b clinical trial. And also simulation results were used to inform dosing selection for subsequent phase 2 clinical trials. So from the phase 1b clinical trial, this anti-FGF receptor 1 and K-loso beta antibody showed some efficacy in reducing liver fat. And what's interesting was this specific receptor is expressed on adipose, not on hepatocytes. So it was proposed that adiponectin released from adipose could have played a role as a mediator. So QSP modeling was employed to determine if adiponectin was a key player in the observed efficacy in liver fat reduction. So this FGF receptor 1 pathway, including adiponectin, was represented in NAFLD-Sim. And the mechanistic simulations were performed with and without incorporating adiponectin effects in simulations. And simulations with adiponectin recapitulated clinically observed liver fat reduction, as indicated in these open red dots here. So the simulation results supported the important role of adiponectin in underlying the observed liver fat reduction, the efficacy of the treatment. And also various dosing regimens were simulated to support dosing selection for the next clinical trial design. The next example was similarly where the QSP model was used to provide mechanistic insight. In this case, GS0976 is an ACC inhibitor, which showed efficacy in steatosis reduction in clinical trials. However, in a subset of treated patients, some unexpected increase in liver triglycerides were observed. So, so QSP modeling was used to really test hypothesis, the potential impact of indirect effect of ACC inhibition that could have attributed to this unexpected outcome. So direct and indirect effects of ACC inhibition mechanistically represented DILIsym. And the mechanistic simulation analysis was performed by incorporating different combination of these direct and indirect effects. And simulations suggested that this unexpected plasma T triglyceride increase was mostly attributed to decreased plasma triglyceride clearance, with potentially some minor contribution of increased VLDL release from hepatocytes. And the fourth example, here QSP modeling using DILIsym was used to predict histological clinical outcomes in NASH patients. In this case, we used zinacrobirg as an exemplar, which is an antagonist of CCR2 and 5. So we obtained mechanistic data from the in vitro studies where zinacrobirg was shown to inhibit activation of hepatic stellate cells and also inhibit the recruitment of monocytes. So these data were used to really represent the therapeutic targets and also derive PD parameters. And we construct developed PBPK model to simulate the exposure. And in this case, the PK exposure, clinical PK data from early clinical trial was used to optimize PBPK model. And then we ran simulations of zinacrobirg clinical protocol using the NAFLD SimPops with different fibrosis stages. And simulations predicted that the zinacrobirg would not provide significant efficacy in improving fibrosis and NASH, as shown in these red boxes here. And after we ran these simulations, the clinical data from Centaur became publicly available, which showed really no improved or no therapeutic benefits of fibrosis improvement compared to the placebo group. So this clinical data validated our simulation results. So this is a good case example where QSP modeling was used to prospectively predict clinical efficacy outcomes in NASH patients, and later that prediction was validated using clinical data. Similarly, this is an example of efficacy prediction in NASH patients, but via different pathway. In this case, FGF19 pathway and its subsequent effects on liver lipid disposition is represented within DILIsym. And in this project, we were able to leverage existing clinical data for other drugs that share the same pathway to calibrate and also validate FGF19 effects on liver fat reduction. Subsequently, we ran simulations of NUM282, an FGF19 analog, and the simulated the serum marker, which were not used in optimization, such as plasma ALT and C4, serum C4, were consistent with clinical observation, and also simulated histological responses such as NAS score and the fibrosis improvement was also recapitulated clinically observed data. The last example here is we used IPF-Sim, where the QSP modeling was used to identify the sub-patient group who would be more responsive to treatment effects of pirfenidone. So within IPF-Sim, we ran simulations of clinical protocols of pirfenidone, and simulations reproduced therapeutic efficacy of pirfenidone, which slowed down disease progression. And in our simulations, we used simulated IPF population, which have variability in the disease progression rate, etc. And also the simulated drug responses represented the inter-individual variability in its efficacy. So we took another step and ran some analysis and identified that among simulated populations, those with greater disease progression rate, were predicted to have greater responses to the treatment, because in this case, the primary outcome was the absolute FBC change from the treatment baseline. We do not have time to really go over all the cases we've covered so far, but on this slide, I have a list of additional cases to give you some flavor of additional cases. So what's listed here is the QSP modeling, utilization QSP modeling to inform clinical design, where QSP modeling focus on FXR pathway in NASH treatment informed optimal duration of the clinical trial to detect histological responses. And in another case where DILIsym was used to simulate a prospective phase 2 clinical protocol, which led to specific recommendations regarding the minimum dose and the target drug concentration and the optimal measurement timing for the liver fat analysis. And here's another example of phase 2-3 like efficacy prediction, where we used IPF-Sim to predict efficacy of a proprietary compound for phase 3 clinical trial. And in another project, we actually used simulated outcome to run biomarker analysis to identify a metric of biomarker exposure that best predicts PD responses, which really helped for the design of clinical trials and endpoints. And here's an DILIsym project where the QSP modeling was used to identify the target NASH population that would show improvement of fibrosis in response to a specific therapy. And lastly, there's a few more examples of mechanistic insight, QSP modeling that provided mechanistic insights, like simulations of a proper compound revealed interaction with the weight gain that offset the potential for efficacy in NAFLD-NASH patients. And then going above liver and lung, we had a project where QSP modeling of platelets in multiple species undergoing irradiation really helped to explain observed species differences in data to provide some mechanistic insights underlying those species differences. And lastly, onto heart, QSP modeling of left ventricle responses to MI indicated that excess anti-inflammatory treatment can actually restrict recovery because it's suppressing some of the inflammatory mediators that are involved in wound healing. Okay, so that concludes my section. And at this time, I'll turn it over to Brett to moderate our panel discussion.
All right, thank you very much, Kyunghee, and thanks to you and Scott and Viera for a very nice presentation that ties together PBPK modeling and QSP modeling and really demonstrates the practicality of the dual approach. So I think we'd start with a few questions. We have a mix of questions, some of which come from our audience, and so I'll start throwing those out. I think we have about 15 minutes to go through and discuss a number of questions, and the first one will be for Kyunghee and Scott. So maybe Kyunghee, you can start, and Scott, you can chime in. What type of pharmacodynamic models are currently being linked to your PBPK models, and do you model cellular level processes in the PD models?
Yeah, so about PD, so it really depends on the disease and target. So We have within DILIsym, I think I present a few targets there in terms of PD effects, mostly impacting has its impact on like liver fat metabolism, and also the immune responses, recruitment of immune cells, and acting directly on fibrosis processes like activation of stellate cells and or collagen synthesis and degradation. So those are a couple examples of targets we or PD effects we represented in DILIsym. And in terms of its representation, if it's a cellular level or not, it depends. Mostly it's cellular level because, for example, like lipid metabolism, that's happening intracellularly. So we have representation of this complex lipid metabolism and different targets among that metabolism pathway. So I would say it's more cellular level, but it depends on the target you're looking at. Scott, do you have anything to add?
That's a very nice description. The one thing I will point out is that, depending on the situation, as Kyunghee said, sometimes we really do focus on the PD. Always we focus on the mechanism of action, the way in which the compound is interacting with the mechanisms of the pathophysiology. And not always, while we may always understand what the PD target is, not always do we understand what that connection between the PD and the mechanism of action actually is, certainly in quantitative terms. And so that is a really critical point, that sometimes we're able to use the QSP modeling to establish some hypotheses about what those ranges are. Other times it is just kind of we'll skip over and into something that's been measured because we're able to kind of focus in on those mechanisms of action. But cellular level almost all the time to directly answer the question.
Yeah, and I think I missed the early part of the question, how it's linked to our PBPK representation. So to address that, it again depends on the site of action or target. If the target or effect is manifest through the intracellular drug exposure, we take intracellular drug exposure predicted from PBPK to derive PD effect. And if it's through the extracellular or cell surface receptor, then we take simulated extracellular drug concentration to derive that PD effect in our QSP modeling.
All right, great. Thank you very much. And I'll a couple questions, additional questions for Scott, and then we'll go to Viera next. So Scott, how do you address the correlations of the variables used to construct the SimPops?
Another excellent question. The answer is, if we understand that two SimPops parameters are interactive, that there is a mechanistic relationship between the two, then we represent, it's incumbent upon us to ensure that that is recapitulated within the simulated patients. And if there's a correlation, then we can certainly allow that to kind of that correlation to be present in the simulated population as well, either as a consequence of the phenotypic description of each patient, or by directly imposing it. I'll give you one example from our DILIsym platform. It's pretty well established and really nice data sets that indicate that body weight, in particular the amount of adipose tissue, absolutely affects the rate of fatty acid release by the adipose tissue, and hence the lipid burden upon the liver and the potential for steatosis. So we absolutely have that same relationship built into our SimPops. So the patients with the largest degree of adiposity have the greatest rates of fatty acid release by the adipose, and conversely, those with less have lower rates. So again, if we understand what the relationships are, we build them in. Once we make the SimPops, we go through and make sure that, are there any actual relationships that were mechanistically imposed versus those that are just kind of surfaced as a consequence, and we'll keep track of all of the above.
Great. Well, one additional question for you, Scott, before we move on to another panelist. How can our audience members learn more about, IPF-Sim and DILIsym? For example, do we have any upcoming workshops or other, types of scenarios?
It seems like that may have been a rhetorical question. Brett may have answered the question for me. Why, yes, in fact, we do have some training workshops for IPF-Sim, for NAFLD-Sim, as well as on the quantitative systems toxicology side for DILIsym and RENAsym. All of the above coming up in September. And so make sure you go to the Simulations Plus website to look for the details and how to register and everything. I'm really excited to be able to kind of walk people through the modeling rationale, the data that we use, even more detail about our SimPops, and our just overall methodology and use of each model.
Great, thank you. So Viera, let's talk about PBPK modeling a little bit. For one question from the audience, while performing PBPK modeling, how do we model the volume of distribution over all the tissues? For example, if the total volume of distribution is known, how do you model it or incorporate that into your modeling process for PBPK?
So with the PBPK model, it's actually a reverse process, right? The total volume of distribution is a result of drug distribution into individual tissues. And it all comes down from having an idea about what mechanisms the compound undergoes, is it substrate for transporters or so, and also looking at the properties of the compound. A lot can be guessed or predicted just looking at the, let's say, ionization, lipophilicity of the compound and so on, and connecting the properties of the compound with the properties of individual tissues in the PBPK model. So it is not simply entering the known volume of distribution, it is more using the properties of the system and the compound to figure out what distribution into which tissues is contributing to that known volume of distribution.
Great, and let's stick with PBPK. One more question there, and this is specifically regarding GastroPlus 9.8.1. How can I add subject-wise age, height, BMI, and weight into the process of PBPK modeling?
This is actually one of the very basic functions in GastroPlus, as you are trying to build a PBPK model for a specific subject that you are trying to simulate, or for a population of subjects. If you are doing population simulation, the program actually asks you, when you are doing a simulation for human, right? The program does ask you to select what population of subjects you want to use, whether you want to sample from Western, Japanese or Chinese populations, right? Whether you want to create a physiology for a healthy subject or potentially subjects with different degrees of renal or hepatic impairment, and also what should be the age, BMI of that subject and so on. So that is one of the basic functions of the program. Actually, I would like to go back to the topic of the workshops that Brett and Scott mentioned earlier, because these sound like fairly basic questions of someone who is new to the program, and just wanted to point out that we do have actually on schedule now introductory workshops for GastroPlus, where these aspects will be covered, guiding you through how to set this up, how to evaluate either the distribution, also setting up the physiologies and so on. So I would suggest that you do take a look at our Simulations Plus website to check the schedule and possibly register for these workshops.
Great. Well, Kyunghee, let's go to you. This is a question still related to exposure, but really from a QSP standpoint. In your experience with the QSP projects you've seen and worked on, how often do exposure-related questions or issues or concerns related to the site of action really take a center stage during that project, or at least become an important component versus, you know, other target-related or disease-related issues?
I would say quite often. I would not say this is only center or only important thing, but if you, as I shared in my intro slide in my Venn diagram, so to predict drug efficacy, you have the right representation of the pathophysiology and population, simulated population, and you need to know the drug exposure at the target. So after we develop a platform of a disease, and then when we move on to really simulate a drug efficacy, well, two important questions we ask is what is the exposure, what's the relevant site of exposure, and then what's the PD effects, how we parameterize those PD effects. And depending on the target, if it's intracellular, for example, we often have discussion around can we confidently predict those tissue exposure or site of exposure at the site of action or not based on the data we have. Sometimes we do have some data to at least indirectly validate those tissue exposure as Viera presented. Sometimes not. Then in that case, for example, if we rely on maybe KP values predicted from in silico model, we use that as a default. But if we know there's some uncertainty in the drug exposure, predicted exposure, we can also run some sensitive analysis or vary that exposure to see if it the predicted efficacy outcome is sensitive to that exposure level or not. So that's one approach that we can take to address the issue. And because coming back to your question, again, its short answer is yes, quite often.
Great. Well, let's go to Scott. This is another question from the audience. Thank you for sharing the great examples in QSP modeling applications to specifically liver and lung diseases. I wonder if there are any other areas or disease areas in which Simulations Plus is interested in developing QSP modeling packages.
Yeah, also a great question. You might have noticed in some of the bullet point examples that there was myocardial infarction, so that's obviously in the heart. Focus on the kidney. We were also mentioning whole body radiation exposure. So we've already kind of began establishing branches of the tree in other directions into other tissues, other disease areas. And I think it kind of establishes proof of principle, or really hopefully establishes confidence in you all that this is actually one of our true skill sets, is to build from scratch de novo QSP and or QST models. And so we have multiple efforts ongoing, some, obviously some models, several versions in, some models currently in construction. And we're happy to work with companies to really identify new disease areas in order to support, obviously some of the sorts of applications ultimately in clinical development of compounds in, like I say, a wide variety of disease areas.
Great. Well, I think we may have time for one more question. So I'll go to Viera. So this is kind of an interesting question that is, again, sort of exposure related, but brings efficacy into the picture. So I thought it's a really interesting question. So for generic development of oral products, there are rare cases wherein bioequivalence is not established, yet efficacy is attained. Have you come across such scenarios and created models for such case studies, which has been approved by regulatory bodies?
That is a very good question. It certainly can be done with the combination of PBPK models if they get linked to the PD models, but I don't recall a specific case where we would have been asked to do this type of study doing the complete link to the PD models. Again, certainly something that can be done if there is enough information available to build a PD model. I just don't recall a scenario where we would have been involved in such study. So it doesn't mean that some of our users were not using GastroPlus for these types of applications. I just don't know.
Great. All right, well, maybe one more question. We have a couple minutes, so I'll squeeze one more in. This one's for you, Kyunghee and Scott. We'll start with Kyunghee quickly. Do customers need to pay for SimPops to be generated and validated for each version or each model, or do the models actually include populations when they're released?
The latter. Short answer is when it's released, it already have the simulated population incorporated as part of it. But that doesn't mean that if they want and customers want any customized SimPops, that can be also done through the consulting services. Scott, do you have anything to add?
Nope, nothing more to add to that.
All right, well, thank you all for a great discussion, and I'll turn it back to you, Arlene, to close it out.
Yes, thank you. Thank you to the panel. You guys did an amazing job. And thank you to our audience. We appreciate your time. And if you've missed any part of this webinar, you will see it on demand tomorrow morning on our website at www.simulations-plus.com. You can also find the workshop registration dates and forms, as well as more detailed information on the case studies and real-world examples presented today. We appreciate your time and have a great day. Thank you.